Okay, so my name is James Davis, and I'm a staff scientist at Argonne National Laboratory. And today's webinar will be on antimicrobial resistance, which we all commonly refer to as AMR, bacteria, and PATRIC. I'm also joint appointed through the University of Chicago's Consortium for Advanced Science and Engineering, which holds the prime contract for the PATRIC project, which I'll talk about in a little bit. Okay, now can I advance? Okay, so today's agenda. Today's talk is the talk is a little bit of a mashup of a bunch of different things. It's, part tutorial and part academic talk. So we're going to kind of be bouncing back and forth. We're going to start with a brief overview of the PATRIC resource, describing what PATRIC actually is, and how you can use it for study. And then the second part, we'll talk about how you can use PATRIC for studying antimicrobial resistance. And we'll do this by demoing the launch of an annotation job. We'll walk through the website for looking at the output of that annotation job. And then we'll describe some of the antimicrobial resistance related annotation features that you get whenever you annotate a genome. The third and fourth part of the academic part, where we're going to talk about an overview of the machine learning based AMR phenotype prediction tools and the metadata collection that fuels the development of those tools. And then finally, we'll end with questions. And I think that today's talk should take about an hour and 15 minutes. Okay. So part one, what is PATRIC? So PATRIC stands for Pathosystems Resource Integration Center, and our URL is patricbrc.org. This is what it looks like whenever you come to the homepage. Can you guys see my cursor? Yes? Hopefully.
Yes.
Okay. PATRIC is a NIAID-funded bioinformatic resource center. NIAID is the National Institutes of Allergy and Infectious Diseases. It's part of the NIH. And it's part of the BRC program, which stands for Bioinformatic Resource Center. PATRIC was initially established in, I think, 2004, and has been in continuous operation since that time, although there have been several mergers and things like that. And at its heart, PATRIC is a data resource designed to support the analysis and comparison of bacterial pathogens, and specifically genomics and their other omics data. But in practice, we have all of the assembled bacterial genomes drawn from GenBank, including collections of other genomes from other places, phage, viral genomes, and a smattering of plasmids and other things. PATRIC is part of the BRC program, and we have sister resources, namely IRD and Viper, which we've merged with. IRD and Viper are large viral bioinformatic resource centers, which are housed or have been developed and housed at JCVI. IRD stands for Influenza Research Database. Their URL is fludb.org. And Viper is the virus pathogen database and analysis resource. And Viper the Viper group has been very active in the current outbreak. And as we are merging together, we're forming a resource called the BVBRC, which stands for the Bacterial and Viral Bioinformatic Resource Center. And that URL is bvbrc.org. And that's currently under development. And so at some point, I think in this calendar year, we will have all of the functionality of Viper, IRD, and PATRIC together under one roof. And the National Institutes of Allergy and Infectious Diseases, their long-term goal is to move all of the BRC resource projects into a single resource. So we will eventually merge with UPathDB and VectorBase, which are undergoing their own merger. So the BVBRC team consists of researchers at the University of Chicago, Argonne National Laboratory, and the Fellowship for Interpretation of Genomes, which is we call FIG, the Biocomplexity Institute Initiative at the University of Virginia, and the J. Craig Venner Institute. We have two PIs. On the bacterial side, the PI is Rick Stevens, and then on the viral side, the PI is Richard Schuerman. So PATRIC can be subdivided into a set of main features, since the talk is about PATRIC, after all. There's a whole bunch of, there's kind of something for everybody in the resource. But the main features that I like to think about whenever I'm describing PATRIC to another person are as follows. The first thing is the database. PATRIC is actually a series of databases, but if you thought about it as one database, that's probably good enough. It is a set of Solr instances that house the data. So what does that mean? Well, the Solr instances have genomes, genome features, genes, proteins, transcriptomic data, antimicrobial resistance data, and a whole host of other things. There's metabolic data, kind of anything under the sun that can be associated with the genomes. In total, there's over 400,000 complete assembled genomes in the database. That number includes phage and plasmids, but there's also over 200,000 bacterial genomes. It undergoes regular updates from GenBank and other sources, and all the genomes that are integrated into the resource get a uniform annotation for comparative analysis purposes. And I think that the database has over 1 billion records, quote unquote, the database. So it's actually a pretty large entity. And that's all housed behind the firewall at Argonne National Laboratory for the University of Chicago. The second main feature for PATRIC is the web interface, which contains web tools for doing comparative analysis of the public genomes and a person's own genomes. So it enables you to view trees, metabolic pathways, compare protein content, view alignments, compare transcriptomic experiments, and it has a whole bunch of other things like circular views for genomes, the compare regions tool, and just a general set of good and well-used tools for doing comparative analysis. So you don't have to have your own genomes to use the resource, or you could use the resource entirely with your own genomes through the workspace, which I'll demo later on in this webinar. The third main feature, which I love, and which is directed at command line users, are the command line interface application and the FTP site. So these tools are designed to enable bulk downloading. So you can take every genome off of the PATRIC website through the FTP site or the command line interface. The command line interface enables also some bulk submission to the well-used services, including assembly and annotation. It also enables bulk download of certain entities out of Solr, so you can get metadata associated with genomes and other things like that. The command line interface can be downloaded from our GitHub site, and we have it can be installed on Mac, Linux, and PC machines fairly easily. So this is a picture down here of the Mac DMG. You literally just drag it to the applications folder. And then this picture over here on the bottom right is the overall structure of the PATRIC FTP site, where a lot of people go to get data really quickly. And the part of PATRIC, the next feature of PATRIC that's probably closest to my heart are the services. The PATRIC services are basically bioinformatic workflows that are usually computationally intensive or contain a lot of individual components that users would take a lot of compute time or a lot of their own personal energy to install. And we handle the compute for these on our side. So a user submits a job, and then they get structured output when the job completes. And on the website, I have a screenshot of it here, and I'll show it to you in a little bit, are the various services. So these include assembly, annotation, metagenomic binning, BLAST, taxonomic binning, which is done with the Kraken tool, similar genome finder, which is MinHash. So a lot of these are high memory or a large amount of CPU time, and we handle all of that behind the scenes on a series of servers. And then when the job completes, this is what the job status page looks like here in the center, and you just get your you get the output back. So these are assembly jobs that I'm showing here, and the services are really popular. The number of public and private this is the number of public and private genomes that have grown, but the service usage can be seen through on the green line. So basically, people are using these services to do assembly, annotation, and a bunch of other things. And then finally, the last thing I'd like to point you to is the help and tutorial pages. There's a very rich tutorial page on the PATRIC website with there are even some videos. There's workshops. We host workshops and webinars like this one where they things get recorded. And Rebecca recently and Anna have been working on establishing a MOOC class for the BRC resource, which has also garnered a lot of attention. And if something goes wrong or you just can't figure something out, you can always drop a message to help@patricbrc.org, where that'll create a user ticket, which will end up going to one of us, depending on the subject. And then we'll get back to you and help you. So the next part, I want to demo just an annotation job and what you're able to do in order to study antimicrobial resistance at PATRIC. So I'm going to bounce out of this PowerPoint, and I'm going to start sharing maybe Firefox. Okay. So I'm going to go to the PATRIC homepage. Can you guys see this? Everybody?
Yes. Yeah.
Okay, cool. All right. The PATRIC webpage works best. The website works best with usually Chrome or Firefox, although I typically use Safari without many issues. So over here on the website, we have a search bar where you can look for data on certain things. And then a lot of the functionalities here in this top, the tabs across the top. The organism tab shows you different organisms of interest where you can look up an organism. The data tab is, again, structured data sets that people are interested in. And obviously help is over here on the right. And the two that I'll use the most in this demo are the workspace tab and the services tab. Services are what I just showed you before. These links will take you to the front page, the widget to submit an annotation or assembly job or whatever. You need to be logged in over here. So if you're going to follow along, the first thing you need to do is log in. And if you don't have an account, you would need to sign up for one. Okay. So I've created a public workspace for this webinar, and I'm going to show you where that is. So we'll practice traversing the workspace. If you go to your home directory, this is what my home workspace looks like. Within the workspace, this is your private data. You can create a folder to hold the output of a job or of an analysis that you do. And then if you go back a level to the total number of work, or let me see here. Go backwards one. So it's possible if you just click on workspaces to get to the higher level workspace. I was in my home directory, which is right here. But these are shared workspace directories that other people have shared with me or that I've shared with other people. Um, and then the last type of workspace environment that you should be aware of are the public workspaces, which are down here on the dropdown. So the workspace that I built for this webinar is here under workspaces, public workspace, and it's this one right here. So I'm going to double click on that. I've created, I downloaded a Staph aureus strain, which happens to be a MRSA strain that I want to show to you today, and that we'll annotate, and that's right here. And I also, in this folder, have the output of an annotation job that I did earlier that we'll walk through as this one is currently running. So I'm going to demo the comprehensive genome analysis service because this bundles assembly and annotation. When you click on one of the services up here from the drop-down menu, it'll bring you to a submission form or a widget that looks like this. They all kind of look the same. This is what assembly looks like. This is what annotation looks like. And here's comprehensive genome analysis. They have info buttons that describe what the service is, and can take you, they sometimes link you out to help and man pages for these. We're going to start with assembled contigs because the comprehensive genome analysis service can do assembly and annotation or annotation, depending on whether you give it reads or assembled contigs. So I'm going to select assembled contigs. If you selected reads, you just give it the read files, and then it would assemble those with one of our assembly pipelines, and then it would proceed into the annotation. But here I'm going to start with assembled contigs. Contigs that I'm going to choose are from that folder in the public workspaces. So what I did here is I clicked on this folder icon, and then it's in the public workspaces, so I'll click on that. AMR bacteria and PATRIC webinar. And then it's the cp029.fna. Okay, and then, oh no, why did it do that?
Give it a second, or
give it a second.
or click the down arrow and see if it's in the top there.
No. You know, I might have to change its file type. That's easy enough. I've been struggling with this the last couple of days, by the way.
Changing the file type is a really good thing to demonstrate.
Yeah, sometimes when you upload something, the system doesn't know what it is. And so if you select the thing that you've uploaded here, and then in the green bar it says edit type, and you can tell it whether it's contigs or a BAM file or a doc file or whatever. So I'm going to tell it that it's contigs, and I'm going to save that file type. Okay, so it's changed it. And now I'm going to go back to the services, comprehensive genome analysis, assembled contigs, and I'm going to pick public workspaces, AMR, bacteria and PATRIC webinar, and then this guy. There. I think that was the problem. Okay, so this is a Staph aureus genome, and the domain is bacteria, obviously. It's, you know, the other options are archaea and viruses. The taxonomy name is Staph aureus, so this thing should autocomplete. And I always tell people that they should get as specific as they can. So I'm doing Staph aureus, subspecies aureus, and then this fills in a taxonomy ID of 46170. Now, if you don't know the taxonomy ID of one of your organisms, you can go to that's the NCBI. That's just the NCBI taxonomy. So you could come to NCBI taxonomy and just do, and it'll give you a number if you click on it. So let me see here. And that's 1280, but I think that got merged with the 46170. So you can always look up a tax ID if you don't know. And sometimes people are working with experimental organisms and they say, well, what do I fill in here if I don't know what it is? And my answer is the most specific thing you can get. So if you know it's bacteria, make it bacteria. If you know it's a gamma proteobacterium, make it gamma proteobacterium. And because sometimes there are taxonomy specific tools that can get run on the annotation side. I'm going to call this Staph aureus webinar. You should call it the most specific thing you can. It will show up on the website like this as a private genome. And then I'm going to create a folder in my home directory, and I'm going to call this webinar assembly. And you should also create one if you're following along. And then there it is. And then select it for the output. Now, oh no. Let's see here. It didn't make my contigs again. There we go. I don't know. This was working. I'm going to try to do this again. I don't know if it timed me out. I apologize, guys. This is seems to be a new issue.
Jim, just to note, I often have problems with the sharing from the public website, and I have been advising people to copy it over to their private website in order to like the reads.
Oh yeah, that's annoying. Okay, well we can do that. Let's just do that.
You can show them how to copy.
Right. So public workspaces. Let's go back here. Yeah, there's more than one way around this problem. We could download it and upload it too if we needed to. So I can, if I move it, it probably goes away. Let me do this. I'm going to copy it to my home directory. And then I created a folder called webinar assembly. I'm going to put it there. Okay, now let's go back and see if we can find it. There it is. Okay. There you go. I think you solved the problem, Rebecca. That's good enough. 1280. Non-label. Up folder. I'll put it in the same folder. Okay. Aha. And now it is submitted. Now, if we watch over here, so there's two ways to monitor the status of a submitted job. The first is to come up here to Workspaces and click on My Jobs. And the second is to go down here to the Jobs page and click on this little button in the bottom right-hand corner. it takes a little while for a submitted job to show up on the job status page, but it will eventually show up here. And when it does, it'll first show up as being queued, and then it'll show up as being running. If it fails, it'll look like this because you'll get a red fail. But it is, I'm going to refresh the page here and see what happens. And here we go. There it is. It's queued. So usually it only takes about 6 minutes to annotate a genome, but that some of this depends on the underlying load across the servers behind the scenes. And it also depends on how heavily any individual is hitting the job scheduler. So, for example, if you're assembling 1,000 genomes on here, and then you add the 1,001st genome, your priority will be lower than someone who's just trying to annotate a genome. So what it tries to do is balance the load from the heavy users with the load from the light users to create a fair environment. Oh, and there we go. Its status just went from queued to running, and it'll eventually go to completed. So the job scheduler handles things in as fair a way as it can. So while that's running, I wanted to share my PowerPoint with you. So I'm going to stop sharing, and then I'm going to start sharing my PowerPoint again. Share. Okay. So I had some still shots in here so that, okay, this is it. This is where I need to be. So while that's running, I wanted to kind of describe what we just did. So the Comprehensive Genome Analysis Service, it's basically the same thing. It's running the assembly service, and it'll run the assembly service if you give it reads, and it's running the annotation service no matter what. And the annotation service at PATRIC is a customized version of the RAST pipeline. RAST stands for Rapid Annotation of using Substance Systems Technology, and that's been around for quite a while. I think since about 2009. And RAST is a pipeline of, I should make this presenter mode. RAST is a pipeline of various algorithms for identifying features in a genome. So the first thing that RAST does is it calls the ribosomal RNAs. And then the next thing it does is it calls the tRNAs. Some of the RAST algorithms are of our own design. So for instance, the ribosomal RNA caller, which finds the 16S, 23S, and 5S genes, that's ours. And then the tRNA scan is a program that was developed by Sean Eddy. So after it gets the ribosomal RNA and the tRNA, it searches for repeat regions in the genome, and then it finds special proteins. These are special proteins are selenocysteine containing proteins and pyrrolysine containing proteins. The reason we look those up with a special program is because they're inserted at a stop codon. And so if you don't go back and correct those, they show up as two broken open reading frames, as one broken open reading frame. Next, it calls CRISPRs, clustered regularly interspaced short palindromic repeats. Then it finally gets down to the business of calling the protein encoding genes. It uses a combination of Prodigal and Glimmer to do this. They're probably the two best gene calling algorithms right now. And then it reconciles the output. They work by having an internal understanding of the codon usage, and it looks for stop codons and tries to assign start codons. And then it assigns the functions of the protein encoding genes that were called using k-mers. And this is the special part of the RAST algorithm. Basically, the way this works is that we have a series of protein 8-mers that are signatures of a given function. And those 8-mers so what happens is that it reads through the open reading frame, finds all of the available, it translates it, finds all the available AMRs, and then looks for the ones that are signatures of a given function and votes. And because this is a K-mer lookup, it's a very fast. And then that's how the function is assigned. And then overlapping genes and other conflicts are resolved. In the final step, the functions, the protein functions that are annotated are compared with other databases that are designed for specific purposes. So we compare the proteins that we call with NDARO and CARD, which are the NCBI and comprehensive and antimicrobial resistant databases for looking up antimicrobial resistance genes. VFDB, Victors, and PATRIC this is a special PATRIC collection for looking at virulence factors. Drug Bank for drug targets, TTD is transporters, and then it also calls human homologs. And these are, I'll show you where these are on the website in a moment. Then it assigns the proteins to protein families, and it finds close neighbors for similarity lookups. So we can check on the job that we have running here. So I'm gonna dive out. I have to dive out of PowerPoint, and I have to switch back to the browser. So I just refreshed the page over here, and the job is still running. So what I'm going to do is I'm going to go back to the job that I did yesterday, so that you can see the output. They're the same thing. There's two ways really to look at the job output on the PATRIC site. The first way is from the jobs page. You can double click on a completed job, and this takes you to the folder where all the job output are displayed. The second way you can do it is that you can navigate to the job output directory that you designated. And so this one for me is in the public workspace because that's where I put it in the webinar. And then here, this is the example comprehensive genome analysis run. And those are the same thing. And when you have the output of a service, you always get this little checkered flag icon whenever it finishes. And so here, this is the same directory of output that we had for the job. And so this is here if you want to follow along. Now, depending on what service you choose, there are various parameters for the programs that you can see if you do the parameter drop down. It gives you the time, the start and stop time, and the job ID. If you ever have a failed job, this job ID is important in order to report an issue with the job. So if you had to report, if you had to contact us about a job that failed, you want to keep that job ID. The parameters tell you what programs were run. Sometimes, in this case, it's just annotation, so you don't see it. But if you were assembling, it would tell you that you assembled with, for instance, with spades and what the parameters were for that and so forth. Now there's two different ways to look at the output. One is that the service will generate a series of flat outputs that you can take and download and use on your own. So, for instance, the annotated genome, you can download that by clicking on the download button here, and that'll trigger a download, and you can look at it. Let's see here. I wonder if it'll come out. There it is. So that's currently downloading, and the annotated genome should probably be a FASTA file. You can look at it with a text. There. So, oh, this is the JSON file. So this is what this looks like. But this is what's read to create all of the other file formats. Okay, go away. Oh, you probably couldn't see that. And then there's also images and stuff like that. I'm going to click on the full genome report. This is probably the most interesting output. This is a summary of the genome that was annotated, and it gives you information. It says it's Staph aureus. This is what we called it. This is organism name. And the report was created yesterday for me because that's when I made this. I'm going to hide it. I'm going to make more space here. And as you walk down through, it gives you information about the various steps of the pipeline. So here, this would give you genome assembly information if there was any to be had, like the L50 and the N50, but it still tells you about the contigs that you submitted for these annotation jobs. So we have one contig in this genome. It was 32% GC, and that's the size. The annotation tells you what features were in the genome. So it had, 2,822 coding sequences. Here's your tRNA and your rRNAs when you did it. And this particular run took 6 minutes and 58 seconds, although the next one might take a little longer because it's still running down here. This little table shows you kind of what was in there at a very, coarse level. So genes that had functions, genes that were hypotheticals, genes with EC numbers, and so forth. This image, which is downloadable, is a circular view with coding sequences, RNA genes, antimicrobial resistance genes, and stuff like that, colored on the genome. I think the one in the center is GC skew. People like to look at these. You can kind of see how the strandedness changes. So this is like a nice complete genome. The subsystem table, pie chart, which everyone loves. Subsystems are essentially genes of related proteins of related function that are clustered together and make up the machinery of the cell. So they break down into various sort of well-chosen blocks. So DNA processing or RNA processing and so forth. The specialty genes are the genes that we're calling that were called there at the end of the pipeline, which included the lookups against CARD and NDARO. Those are actually BLAT searches, which is basically the same thing as BLAST and from a functional perspective. Drug targets, transporters, and virulence factors for easy lookup. This I'll talk about more in a little bit. It gives it a prediction of what this organism is resistant to in terms of antimicrobial resistance. These are done with machine learning algorithms, and I'll show you. I'll show this to you in the sort of academic portion of the webinar. We've got antimicrobial resistance genes that were found. I think if I shrink this will render correctly, and we also have a tree of our organism in the context of the other organisms, and the NEWIC file for that tree can be downloaded as well. So, but this is static. This is the static report of one genome that was generated by one job. That's not where the full functionality of PATRIC actually lies. The full functionality of PATRIC lies with the comparative analysis environment. So if this was your private genome, which it will be after you annotate it, you want to be able to look at it in the context of the other genomes. And so what the system does is that it indexes your genome against the other genomes in the resource so that you can do comparative analysis. And so if you want to see your genome in the context of the other genomes, there's two ways to do it. The first is that you can click this view button whenever you're in the output directory. So if I click that, it takes me to the landing page for the genome. The other way to get there is to go to the organisms page. This was a bacterium, so I'll click on all bacteria. Genomes. This is every bacterial genome, so this takes a second. And then you can filter for public and private. And so I've got 2,300 genomes in here that are private. But I should be able to find
I think I can't tell because it's too small, but I think you've misspelled Staphylococcus. But maybe I'm wrong.
No, I had it right. Maybe I got the filter wrong. Public, true. Ah, well, I'm not seeing it here.
Try going up to workspaces in my genomes.
Yeah. Oh, my genomes. Look at that, Rebecca. And I should be able to find it in here. Hmm. Oh, those are groups. Well, that was a fail,
Those
but I
aren't
know how to.
those aren't groups, those are shared, I
Oh,
believe.
oh, maybe that's why it was hard to see because it's a public. I generated a public genome. Oh, I think that's why it's messed up because because I sent the output to the public shared space. It's not showing up as a proper private genome. I didn't realize that was going to mess me up so badly. So we're going to go back and we're going to view it this way. This is the landing page for the particular genome that was annotated. So it would have shown up, I think, in the genome browser as a private genome, but I think that I ruined that by annotating it into the public workspace directory. And I think as a result, it couldn't see it, but it did get indexed against the Solar database. And the center pane here takes a while to load, so we might not get to see it all. But what it does is it's basically a rundown of the same things we saw with the Comprehensive Genome Analysis Service. So the table of hypothetical proteins, which you can click on and get, or functional assignments, virulence factors, antibiotic resistance genes, and different various features. If it were every public genome also has a landing page like this. And when it is a public genome, it'll give you information about the isolate and stuff that had been submitted to GenBank. And so if it had an isolation host organism that it was isolated from, or metadata about that strain, that would all show up under here. And so those are pretty useful for trying to find organisms that have particular features that you like. I'll come back to this page, but these are predicted phenotypes. Predicted phenotypes are made using the machine learning algorithms, which I'll describe later. So we'll come back to this, but it's trying to say that it's resistant to these particular antibiotics. The phylogeny tab gives you phylogeny. The genome browser shows you the genomes. Actually, that'll show you features of the genome. So this is, you're able to actually walk the genome through here. I'm not going to load it this way. I'll show it to you in a minute from the features page. So here is every feature that was called in the genome. And here's all those specialty genes. So these are the genomes that match PATRIC, that match the PATRIC, CARD, and NDARO data sets. This might take a second to load. So here we go. And I can filter this for antibiotic resistance. And so here are the genes, and then the source. So if we wanted to look at CARD first, the match is based on BLAT. CARD, it'll give you the percent identities 100%, and this is gyrase, which is usually involved in resistance to fluoroquinolones, like ciprofloxacin, for instance. Here's the polymerases, which are involved in resistance to rifampicin and so forth. So you can go down through here and look for your antibiotic resistance genes. And you can compare the sources of those annotations. And now, when you're looking at this, you should take it in context for what it is. For instance, the beta lactamases which confer resistance to the beta-lactam antibiotics like penicillin, for instance, their annotations can get very specific down to the level of the single nucleotide polymorphism. And so when this is compared to NDARO, this is based on protein similarity. And the proteins, the product here is the function, the proposed function for the protein. So what you might see, for instance, is here, class A beta-lactamase. You'll get the EC number for a beta-lactamase, and then the family that the beta-lactamase should belong to. So this is apparently Blasi family. But you'll notice that it's not a perfect match to the NDARO collection. And what that ultimately means is that the very, 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 very specific SNP-based nomenclature breaks down because we are looking at antimicrobial resistance at the protein level here. And so our recommendation to someone who is diving that deeply into the SNP level changes relating to antibiotic resistance are to go back and double check against CARD and NDARO to see how this holds up. What we can offer though to the user is, if we say it belongs to one of these families, it'll belong to that family. And the aminoglycosides are particularly bad. So there are clusters. The annotators have created clusters of these. Make that a little bigger because it's starting to get small on us. Oh shoot! This is fine. They've made clusters of these that can get sort of large because they can create polyphyletic groups and stuff. So this is where you get your antimicrobial resistance genes, and this is where you can decide which one you want to look up against, or how you get the information related to them. So one of the genes that I was interested in, because this happens to be a Staph aureus strain, is MEK-A, which confers methicillin resistance. So here's MEK-A. This is good because we've caught it with CARD and NDARO. Notice that they're the same thing. This is their protein identifier in PATRIC. They're the same protein. Here's the repressor, which is right upstream of it. Now, if you wanted to look at that specific feature, you can click on the features button over here, and you hit the feature landing page. Now, the feature landing page is pretty cool because it gives you information about this, and it also takes you to the source IDs for the genes elsewhere, or the proteins elsewhere, and you can look at that specific region. So here's MEK-A, here's MEK-R1, so that we're in this element. And the machine learning algorithms, I won't get the mouse over here. Oh, there it is. Oh. There we go. Oh. We'll find it this way. So you can also look on the genome browser, which enables you to walk the genome. And for this particular gene, it highlights MEK-A because that was the feature I clicked on. And then you can see that the classifiers, the machine learning classifiers, are saying that this is a methicillin resistance predicted region. And I'll show you how those work in a minute. And then you can just click and scroll, and you can literally walk the whole genome. Now, we also have the genome browser tracks for publicly available genomes. And if they came out of RefSeq, you'll get a secondary track of what RefSeq called it at the time it was integrated into the resource. So you can also look that up as well. And that'll have the accession number for that particular gene with the ability to look it up. So now going back, protein families. So if you went back here to this particular feature, you click on the feature page, it'll give you the protein families. That's one last thing I wanted to show. The global family is allowed to extend beyond Staphylococcus, but this is Staphylococcus specific. And so since this is MEK-A we're looking at, clicking on the protein family gives us nearly all of the MEK-A genes for all of Staphylococcus. So if you wanted to create groups or build trees or do anything like that, or build alignments from common, genes of the same function, you can do it through the protein families. And so that's basically all I wanted to demo with respect to the PATRIC website, and getting into the AMR functionality of PATRIC. There's a lot more here under the hood, but for the period of time that we have for the webinar, that's what I wanted to show. And I would encourage all of you to sign up for Rebecca's webinar, which is more of a comparative analysis style, which will be more of a comparative analysis style webinar, which I think is in May. Is that May or April, Rebecca?
June 1st.
Oh, June.
I believe it's June 1st. And we will, I was thinking more of going through finding public data and comparing it within the public data. Also, we have the MOOC course that I put in the chat. Jim Ahmed has asked, and I did answer him, but you probably can go into more detail on this. When you run your genome on PATRIC and are you ready for questions? Sorry, I thought you were.
I can take some questions now, and then I'm going to jump into the AMR classifiers after
okay,
that.
Just very quick.
This is a good point to do that.
Just quickly, he wants to know what the differences are when you run the same contigs on PATRIC, and if you take it over to RAST and do it.
They should be nearly identical. They actually call the same underlying set of algorithms. There are a few legacy features that you'll get at RAST, which include the comparative analysis environment over at RAST, which is a little bit different than what we have at PATRIC. But they're calling the same server. Then over on the PATRIC side, I'm not sure if you get specialty genes over at RAST or not. There's a couple of things that are done on the PATRIC side that enable us to look at that genome against PATRIC. And so the short answer is that they're almost identical. And unless you're in some like a fringe of annotation situation where you're looking at something very specific to RAST, you'll get the same thing at PATRIC. And then if you run it at PATRIC, then you get the comparative analysis environment of PATRIC that comes along for the ride. If you're very familiar with RAST, there's a way to actually customize a RAST job by turning on and off certain annotation algorithms. So you could turn off the tRNA caller, for instance. You can't do that at PATRIC. And then if you were to customize a pipeline at RAST in that way, then it would be kind of incompatible over here. So those are the main differences.
Jim Ahmed asks, can we use GFF file analysis in PATRIC? And I can say that the answer to that is, alas, no, because you have to submit contigs for annotation, and
we start with contigs, but somewhere you're testing me because it's been a while since we've written a paper on RAS. Somewhere deep in the functionality of the command line interface, I think that it's possible to do this either at PATRIC or RAST, because people have asked that question before, but I don't know the answer offhand. And I actually don't know if the little stub of code that did that is still alive and visible in a place where we could point him. So offhand, I would have to say no.
Okay. Any other questions, anybody?
And that's true of the GenBank file too, that people ask about some of annotating GenBank files as well. Okay. All right, then I'm going to move on to this is sort of the talky part of the webinar. So one of the things that I showed you but didn't explain were these the phenotype predictions that corresponded with an annotation job. So what are these? These are machine learning based predictions. They're from machine learning based classifiers. So part 3, machine learning based AMR prediction. So most people traditionally predict antimicrobial resistance phenotypes using known AMR genes. So if they see the appropriate set of mutations and the DNA gyrase, they'll say that's, or the topoisomerase, they'll say that that's Cipro resistant, and they'll make the assertion based off of the SNPs or the presence of a given antimicrobial resistance gene, depending on whether it's SNP driven or horizontal gene transfer driven. This is well established and it works well, and there's a bunch of resources that do this already. That's not the direction that we went in, especially because PATRIC is protein function centric. The way that we do it is that we're using machine learning algorithms to make a genotype to phenotype prediction. So machine learning has some advantages and disadvantages. Some of the advantages are that we don't need to know the gene content or the SNP patterns. The prediction is made directly from the DNA sequence of the genome, which I'll explain momentarily. We don't need a reference database of well-curated SNPs, which turns out to be labor-intensive, or AMR genes. And sometimes the machine learning models can capture complex genetic relationships that are interesting and provide us with the ability to use them for gene discovery as well, or function discovery, I should say. There's a couple different ways that you can design a machine learning algorithm for predicting susceptible and resistant phenotypes. But at its core, what you're trying to do is you're trying to take, so if you're building a classifier for predicting, say, susceptible or resistant genomes, what you're doing is you're taking a diverse set of susceptible genomes and a diverse set of resistant genomes, and you're letting the machine learning algorithm or the classifier parse these two sets of genomes to find the important aspects of one or the other so that it can make a prediction. So it's just essentially a fancy voting scheme. The ensemble machine learning methods work pretty well for doing this. We've successfully built machine learning models using AdaBoost, Random Forest, XGBoost, and a variety of other methods. Other groups have used set covering machines and support vector machines. The deep learning methods can work, but you have to set up the experimental design so that they don't gobble up a lot of memory. It has to go on a GPU. So, and we've actually published several papers at this point trying to figure out how to do this best. And so when I'm wearing my PATRIC hat, I'm actually the bacterial services lead for the project. And so we're always trying to figure out how to build new and interesting and incisive services that people can use in order to get more value out of the data that they're submitting to the resource. And so this was one of those projects. There are disadvantages in using machine learning classifiers to do this, and they're visible in the website if you look. This is a work in progress. They require a lot of genomes, and the genomes need to be sort of diverse. They need to represent like what you might expect in nature. They require antimicrobial susceptibility test data. So you need to be able to find genomes that are linked to some laboratory test. And there's machine learning considerations like balancing, preventing overfitting, and things like that. Another way that you can design one of these is as a regressor. So in this case, one of the projects that we also did, this is not in PATRIC yet, but it needs to get put into PATRIC, is predicting the minimum inhibitory concentration. And this actually is possible using regression. And we've written a couple of papers on that as well, where we took well-curated data sets where we had antimicrobial susceptibility test data, and we were able to predict the minimum inhibitory concentrations of those genomes. So the current state of the classifiers in PATRIC is that the MIC prediction is not, minimum inhibitory concentration prediction is not yet integrated, but susceptible intermediate resistant prediction is. But we're at the point where we need to update that. We kind of got stuck in the queue behind all of the COVID work that's been happening lately. If you were to take all of the data in PATRIC right now and build one large classifier for it, which we've done, in order to predict susceptibility versus resistance, and you can make classifiers for predicting susceptibility, intermediate and resistant phenotypes, susceptible, non-susceptible, intermediate susceptibility. Like there's a bunch of different ways you could look at this, but the easiest is susceptible versus resistant. These are the organisms for which you can make a prediction right now, and then across the x-axis here of the heat map, we have the antibiotic abbreviation, and then the coloration is the F1 score, which is the harmonic mean of precision and recall. This, you could read this as accuracy if you'd like. They're almost the same thing. So the darker greens are the more accurate classifications, and the lighter greens are the less accurate ones. So this is all of the species munched together to build a classifier that have more than 75 susceptible or resistant genomes. It's the input features are nucleotide 7mers. So you chop up each assembled genome into 7 nucleotide segments, stepping by one over each genome, and you build a matrix for this. And the particular algorithm that we used in this case was XGBoost. This is a pretty good model. You can see that it's working on a lot of organisms. And then the all columns here are the averages for the columns and the averages for the rows. We have less minimum inhibitory concentration data right now, but this is what a 7mer model using XGBoost as a regressor looks like for this. And this is the F1 score within plus or minus one twofold dilution step. We bound the accuracy by one plus or minus one twofold dilution step because that's the limit of resolution for a lot of the automated MIC measuring methods. This is kind of a busy figure, but across the bottom we have the ability to predict MICs for Acinetobacter, Campylobacter, Enterobacter, E. coli, Klebsiella, Mycobacterium, Pseudomonas salmonella, and Streptococcus right now. So that's where we're at with the current state of the art for predicting phenotypes for organisms. There are a whole variety of other metadata categories that can be downloaded out of PATRIC, and you can download the antimicrobial resistance phenotypes, which I'll show in a minute, or you can download other things like the host organisms, or the human body site, or the environment of isolation, or the geographic location, and tinker with those categories as well, which we've done on the side, but are beyond the scope of this talk. So this brings me to the fourth and last part of the webinar, which is the antimicrobial resistance metadata collection. At PATRIC, we have been collecting antimicrobial resistance metadata for a really long time. And what do I mean by that? Well, in order to build machine learning models or do GWAS studies or comparative analysis, a rich comparative analysis, you actually need to know the phenotypes that correspond to the genomes in the database. This is the metadata, or these are the metadata. These are sometimes called antimicrobial susceptibility test data. And what they are laboratory-derived antimicrobial susceptibility tests results paired with the bacterial genomes. They're typically displayed as susceptible, intermediate, or resistant calls. Sometimes you see non-susceptible, reduced susceptibility, and things like that. We also have a fair amount of minimum inhibitory concentration data, which we kind of showed there a minute ago. And we actually have some zone diameters for disk diffusion assays and things like that. We've been collecting these data out of the literature since at least 2014. We occasionally sync up against the NCBI data set, but we're a superset of those of the NCBI data because we've been pulling the data out of the literature. People will publish genomes and they have to publish them to SRA, ENA, or GenBank, but they don't technically have to publish the AMR metadata, and so you have to kind of dig through the supplements of papers and things like that. The entire collection exists on the FTP site, and it's also mirrored on the BVBRC FTP site, but they're the same file. And so at this point, the main sources are the literature, the NCBI antibiogram template, and various NIAID projects, NIAID-funded projects where we've integrated the data. And the characteristics of the data collection are that we have 67,000 genomes with some form of antimicrobial susceptibility test data. They cover 39 genera and 89 species that are named. I don't count the .sp species. There's way more if you count those. It covers 128 antibiotics. Some of them are like compounds and things that aren't really traditional antibiotics or topical antibiotics or veterinary antibiotics. But there's 128 compounds in there. We've got 324,000 minimum inhibitory concentrations and 356,000 susceptible, intermediate, and resistant calls. Approximately 20 measurement methods. By measurement methods, I mean the BD Phoenix, VTEC2, E-test, disk diffusion, those sorts of things. And over 218 publications that have been scoured and sourced for the data. So we maintain a data sheet of, we're planning to write a review article on this, which is why this is a supplemental table right now. But we maintain a data table of all of the publications that we've sourced for the PATRIC resource. And we're going to make that publicly available in the next month or two. Our metadata fields that we try to populate are very similar to the NCBI antibiogram template. We're trying to get the genome, the taxon ID, the antibiotic, obviously, and the phenotype, the measurement sign, the typing method. If it's a susceptible, intermediate, and resistant call, we want to know the standard. If it's a particular machine, we want to know who the vendor is, what year the standard was that was established, because sometimes the standards change for calling susceptible and intermediate and resistant genomes, and sometimes the various standards like CLSI and UCAS, sometimes they differ, and so you need to know those sorts of things in order to work with the data. Currently have the most data. This is a histogram of the number of genomes with antimicrobial susceptibility test data. And we currently have the most for tuberculosis and salmonella, and then it sort of drops off rather quickly in the other organisms. But these are starting to populate. So we're starting to get more Shigella, we're starting to get more Enterococcus, more of the Klebsiella and Staph the Staph aureus. There's Staph that isn't aureus and Klebsiella that isn't pneumoniae and stuff like that. So we're starting to get a lot more data for diverse organisms. The study design largely dictates the number of antibiotics that are tested for each species. And so we have a pretty large standard deviation on the average. That's what the error bar is here. But, it's not uncommon to see between 5 and 20 antibiotics tested for each organism. And that also depends on the species. You'll get more antibiotics tested on the gram-negative enterics, like Klebsiella and E. coli, than you'll typically see for Mycobacterium tuberculosis, because there's only a set number of drugs that work on tuberculosis, and they're usually different. This is a beautiful figure that Rebecca built for this project that shows the number of genomes per country of isolation for the top four organisms. So from left to right, we have Mycobacterium tuberculosis, gonorrhea, Salmonella enterica, and Strep pneumoniae. And what you can see here is that there's a bias in which countries are sampling which organisms. That depends on sort of the burden of disease in certain countries, and also the priorities of various funding agencies and monitoring agencies. And so, for instance, Salmonella, we have a lot coming from the US and Canada. And then for enterica, these are usually Typhimurium strains for US and Canada. And then over here in the Indian subcontinent, that's a lot of paratyphi and typhi strains from various outbreaks that they've suffered through. For tuberculosis, we got a lot of sampling out of Asia, the Indian subcontinent, and certain very specific African and South American countries, and it's almost non-existent in the United States. That's not to say patients don't get it. It's just that they're not sequencing and submitting genomes with AST data for these. And then, of course, strep pneumoniae is a little bit different from the others as well. And so there's a study there are skews relating to the study bias there are skews relating to outbreaks, for instance, with Salmonella enterica, and there are skews relating to the priorities of funding and monitoring agencies. So if you're to use the AMR data set that exists in PATRIC, you want to be aware of these up front. This heat map shows the sampling by antibiotic for each organism, and this is genomes where there are more than 50 of susceptible intermediate or susceptible or resistant genomes. And then the scale is capped at 500, so it's not so it's readable. This is all of them by antibiotic. So you can see that we're starting to see a lot of more interesting stuff filling in. And then these are the minimum inhibitory concentrations, and this looks a lot like this tracks very closely with the accuracy of the models that were built that I showed you earlier. And there's a couple other things I wanted to show before I stop for questions. This is the actual distribution of particular antibiotic resistance genes sometimes skews away from what you might expect in the database. So the fraction here on the left is the fraction of a given beta-lactamase or TET resistance gene for the given organism in the collection versus the entire database for PATRIC, which subsamples the AST collection. And so if you look at the first line of the table here for class A beta-lactamase TEM family, Klebsiella pneumoniae is way oversampled relative to the database for TEM beta-lactamases, and that's also true of the SHV family and the CTX family. And that's because a lot of the Klebsiella studies that have contributed data have been very focused on extended spectrum beta-lactamase producers. And we see a little bit of an oversampling of tetracycline resistance, TET-A, in the Salmonella set because a lot of these were FDA and USDA genomes that where tetracycline resistance is an issue. And so these are some of the inherent skews that exist in the data that are important to consider. So I'm going to leave with the last two slides I'm going to show are what we're working on currently. We've been building models that don't require an entire genome in order to make a prediction. And we just published one in PLOS Comp Bio in the fall, and we have one that's under review right now that I hope we can get published. And so what we're finding is that a subset of genomes or a subset of the genome, a particular part of the genome might actually work as a good proxy for the entire genome. And this is a plot of a whole chromosomal alignment showing the accuracy of each antibiotic over a particular region for Klebsiella, tuberculosis, and Salmonella. And what we're finding is that so for tuberculosis, the really high peaks are the ERM-A and B genes, the RPOB gene, and the INH, the isoniazid resistance genes. So they're creating the peaks, but that we get a high residual background accuracy even when we only use a small part of the genome. So that's pretty cool. And I obviously don't have time to go deeply into depth with those, but if you wanted to check those out, this one's published and we're getting close with that one. So that brings me to the end of the talk. If you want to cite PATRIC, we would encourage you to, if you're using PATRIC to cite us, and that's our current NAR database issue paper. And I would like to acknowledge all the people who have poured energy into this project over the years, including the teams that I mentioned up front. Oops. And for the AMR metadata collection, we have we've been curating that collection primarily with NIAID funding, but also recently with DARPA funds for building models for a very specific purpose on the DARPA side. And so Margot Van O'Fellen has been doing a lot of the metadata curation lately, and Marcus and Daria made the models that that I showed you earlier. And so with that, I can take questions. Oh, and this is the group that works with me. These guys are great. Marcus is on the left, and Daria is the second from the right here. So are there any questions? No?
Well,
Crickets?
there have been several in the chat, but
Okay.
but
Should
we've
go through them, I
addressed
guess.
those. I've addressed
Okay.
those.
Okay.
If
Well, Rebecca, do you have any questions?
I think it's pretty great. One of the things that you didn't show that how it looks, how you can see how it's viewed, like on the genome page, for specific genes, you can see where the classifiers fall. That's one of the cool things that Jim has done. So often you'll find that these classifiers, when you look at them,
Yeah, like here.
yeah, you can see that, but it's also on the feature page too. You can see the surrounding genes and stuff. Here's a question I have. Are we ever going to bring the classifiers
Oh, you
into
mean
the
like here, where these sprinkle all through? This is the
Yes, I
SCC
love
mech
that.
element. Yeah,
Yeah.
and these sprinkle all through. They're not always right, because the machine learning model doesn't know anything about the underlying data. So sometimes you'll see one that's along for the ride. So I think in these two, one of them is methicillin resistance, and the other one is tetracycline resistance. But I think that's because there's a strong association with the SEC mech element and these other forms of resistance. So those are sort of they're evolving, but they're really neat because they draw your attention into an area that is interesting.
And also it gives you a sense, you can feel the correctness of it or not if you're seeing them all classifying around known genes
Right.
for that.
Sometimes they pile up into an intergenic region, and I was building one time. I can't remember what antibiotic it was, but it turned out to be the sort of the integration site for a particular element. And the susceptible ones had the integration site intact, and the resistant ones had the integration site not intact. And so that was the main feature. And it wasn't the antibiotic resistance gene, it was the actual integration site. And so there's sometimes deeper meaning in what the algorithm finds that you wouldn't initially think of, or that might not have gotten annotated by our system.
You do have a question if you open up the chat. Do you know how to do that?
Okay.
Does Patrick have an option to predict the structure of operons or genes associated with metal resistance?
We don't have anything specifically for metal resistance. We also do not specifically offer the ability to predict an operon, but in the bacteria, you can kind of infer the operon structure with the genome browser or the compare regions tool. And the compare regions tool is kind of neat because it colors the genes of a given region based on their similarity. And so you can compare many genomes. Let me see. I have a picture of that. Hold on a second. You can compare many genomes. Here it is. It's this down here in the bottom left corner. You can kind of compare genomes this way, and you can see the operon structure sometimes pops out because it decays with phylogenetic distance. But we don't have a specific algorithm for calling operons.
We should also remind people that we have some more seminars coming up on the respiratory pathogen series. On
Ah.
April 20th, we have RSV, pneumoviridae, and viper. On May 4th, we have aspergillus and fungi DB. On May 18th,
cool.
we will have enterovirus, picornaviridae, and viper. And we end up finally with bacterial respiratory pathogens and PATRIC on June 1st, just so everybody's aware.
That sounds like a hot lineup. Do we have any speakers locked in for that?
I'm sure they do, but I don't see it in the list. We have
Okay.
another question. Does PATRIC have an option to predict the structure? Oh, we just did that. Okay.
Yeah.
Did I say metal resistance?
Yeah, but we don't
Okay.
specifically have metal resistance, although
We
sometimes,
sometimes do in the subsystems. So if you
yeah,
go into
exactly.
subsystems and look at virulence, you'll often get metal resistance. Now, let me point out that PATRIC subsystem is not as up to date as RAST subsystem. So if I was really interested in this, I would annotate my genome at RAST and look at the subsystems there. Also, they open a lot easier. Right now, we have some database issues that we're trying to resolve that impact subsystems, and our pathway things make it very slow and hard to open. RAST does things a lot more quickly. So if you're really interested in that, do it in RAST, except you
Yeah.
won't have the comparative analysis capability that you have in PATRIC.
Rast has less things tied to other things. And so sometimes it can be more responsive. The other thing, there are other tricks that you can do for metal resistance too. You can search for a given metal, like if,
Right.
you know, in the feature page and see what genes pop up from their functions, and then move from there on, into the genome browser or something where you can look around. The other thing that you can do is, if you know EC numbers of specific metal resistance genes, you might also be able to search by the EC number and see if there are particular pathways.
Also,
That
even from the genome landing page, that text box that Jim was showing you at the beginning, that can be very powerful. I had a user yesterday ask me, do we have any genomes that have been subjected to
Here.
UV mutagenesis? And when you're on the
Oh, I have
genome
no
table,
idea.
oh yeah, it was
We
easy.
do?
I put it when you're on the genome table in the text box at the top, I put in UV, I think it was staph that they were interested in, and it came up with like 5 genomes. And when you went in and looked at the notes associated with that genome, it said they had been subjected to mutagenesis
Oh, get out of here. That's
specifically.
cool.
So you don't be afraid to use that text box to drill into things. It's looking for specific words. So if you're interested in metal resistance, use that particular word in there, and you can find it there that has some genomes that are associated with that.
Yeah, and there's another thing to keep in mind as you use that search box, is that it's searching through the entire record for that genome. And so sometimes you'll search for something and it'll bring up an answer that you didn't expect.
Yeah.
And that's because somewhere there's some linkage or a word that it catches, which might not be, the name of the organism or something like that.
Here's another question that both Anna and Ahmed have both asked, sort of. Anna asked, does it have a feature to present antibiotic-resistant genes from several isolates in a heat map? And yes, we do. You can use the protein family sorter. And if you have the protein family ID, you can filter the protein family sorter and show specifically that. And that is included in that MOOC course if I've shown people how to do that. Not so much with antibiotic resistance genes. I show how to do it with the lipopolysaccharide genes from Brucella, but you can use it for anyone. And that's a common question that we get at PATRIC. So it would be worth your while just to sign up for that course, and you can even cherry pick the different things you're interested in.
Yeah, and the other thing is that there should be a tutorial page on how to use the heat map.
There is, but I think that the course goes in more detail because I was able to expand it. But yes, there are tutorials on how to use the heat map in the protein family sorter as well.
Yeah.
Any other questions?
Well, this was a great time. I had a blast. I hope you did too. I hope I didn't anesthetize anyone.
I've put different emails. If you're
Okay.
interested in getting in touch with us, help at patrickbrc.org is in the chat. And I also, well, gave my email. Oh, and there's Jim's email. So plenty of emails.
Yeah,
Yeah.
we'll get it sorted. If you need help, we'll get you into the right place.
Yeah, and let us know when you have problems, because if people don't tell us, we think that everything's going swimmingly. So we need to know in order to fix things to make it work for you.
Yes. All right. Well, that concludes our webinar on antimicrobial resistance and bacteria and PATRIC.